So now it's my happy duty to introduce Peter Agre. And Peter is a colleague and a friend and is our next speaker. He came to Johns Hopkins in the 1970s for medical school and discovered a love for both biomedical research and for diseases that affected developing countries. He spent most of his professional life at Johns Hopkins with a short detour to Duke University. He started at Hopkins with a postdoctoral fellowship in cell biology, and then he joined the faculty of the School of Medicine in 1984. In 2008, he was named University Professor and Director of the Malaria Research Institute at the Johns Hopkins Bloomberg School of Public Health. Peter's research in red blood cell biochemistry led to the identification of the first known membrane defects in congenital hemolytic anemias. He studied hereditary serocytosis, and he produced the first isolation of the Rh blood group antigens. In 1992, his laboratory became widely recognized for discovering the aquaporins. These are a family of water channel proteins found throughout nature, highly conserved, and responsible for numerous physiological processes in humans, including concentration of urine in the kidney, as well as secretion of cerebrospinal fluid, aqueous humor in the eye, tears, sweat, and release of glycerol from fat. Aquaporins have been implicated in multiple clinical disorders, including fluid retention, bedwetting, brain edema, cataracts, heat shock, and obesity. Water transport in lower organisms, microbes, and plants also depend upon aquaporins. His work changed the way we think about how cells work and our understanding of human health and disease. And for this work, in 2003, Peter received the Nobel Prize in Chemistry with Roderick McKinnon of Rockefeller Universities for, quote, discoveries concerning channels in cell membranes, unquote. As president of the American Association for the Advancement of Science, Peter led science in diplomacy missions to Cuba, North Korea, and Myanmar. His honors include elections to the National Academy of Sciences in 2000, the Institute of Medicine in 2005, the American Academy of Arts and Sciences in 2003, and the American Philosophical Society in 2004. He has received 16 honorary degrees from international universities and was also appointed Commander of the Royal Norwegian Order of Merit by King Harold V. Peter and his wife Mary have 5 children, live in Baltimore, but he's really a citizen of the world. So, Peter, please come.
Well, thank you. It's a pleasure to be here. This is not my first trip to Berlin. I first came to Berlin in 1966 as a high school student. The world was different then, Berlin was different then, but it was an amazing experience. And I think at that time, the whole world said something we still agree with, that we are all Berliners. Wir sind alle Berlinerinnen. Is that German? So I'm going to tell you a story. Not just about science, but how science opens doors, and it has something to do with youth. Youth is an essential ingredient of science. Scientific opportunities are essential, and the doors that open worldwide are a benefit that we can make use of because of our scientific friendships. So this is me. This is actually a few years after I was in Berlin. Had more hair back then, but I was a young scientist leaving my home state of Minnesota to come to Johns Hopkins to save the world, or so I thought. Hopkins had already established a very large reputation for scientific excellence in the global health arena. And like other young scientists, I knew nothing. I had passion, I had enthusiasm, but the only way forward is to have mentors. And I was fortunate enough as a medical student to fall into this laboratory run by these 2 gentlemen, Pedro Quattrocasas in the pink shirt, Spanish-born biochemist, and Gianfredo Puga, the mustachioed fellow on the left, who ran an amazing membrane biology laboratory. And their passions extended not just from the science, but to way beyond the science. And Pedro had pioneered a technique known as affinity chromatography for purification of molecules and did the first purification of the insulin receptor. And Gianfredo, a handsome Italian, decided he would solve the most important problem any medical scientist in Italy could solve, the molecular basis of femininity. And he succeeded with the isolation of the estrogen receptor. And they were debonair role models in every way. The rest of the lab was a little less debonair. We looked sort of like a motorcycle gang, actually, my wife tells me. And we had people from really all over the world pursuing scientific passions on interesting and important medical problems. Surprisingly, the people came from backgrounds which were oftentimes quite unfriendly, and this is the opening of doors. One summer we were joined by these two young men, the laughing young men with beards in the front. Would you be interested if I told you on the left, Naji Sahioun was a Palestinian, grew up with the hatred of Israel and suspicion that American Jews were Zionists. And on the right is Marvin Ira Siegel, son of an Orthodox rabbi from Brooklyn, New York. And they worked together for 2 years, became the best of friends to the ends of their lives. There's something special about science and scientific friendships. Now, as Mike mentioned, we worked on the water channel proteins, and I'll talk about them briefly, mostly as a segue to talk about opening doors. Water, of course, is an essential component of all cells, a major component of all cells and all tissues of our body. And most of the time, water moves with smooth transition from one body to another. We can make spinal fluid, tears, sweat, we can concentrate on urine. But when water is in disequilibrium, it can be quite dangerous. And many medical problems involve the disordered transport of water. Our laboratory, really by sheer serendipity, identified a protein which turns out to be the molecular water channel, the plumbing system of cells, allowing water to go across cell membranes. And the key experiment, a very simple experiment shown here, shows the presence of a water channel in the right egg. This is a frog egg, a cell in which we express the water channel protein. So this one has the protein present. And to the left, we have a control cell. And in culture medium with isotonic buffer, there's no osmotic gradient. So the idea of aquaporins is that they allow osmosis to occur very rapidly. So transferring these together to distilled water, an amazing and immediate difference is noticed. The test oocyte is swollen and exploded, producing much jubilation in the laboratory. This is Greg Preston. I actually took this photo of Greg 3 years after the discovery. He was still celebrating. And this opened up some opportunities for us because we were a small laboratory and we felt that the interest in this was much bigger than we could really compete with. So we teamed up with other scientists and became really acquainted with a world community of scientists quite early. There are a number of aquaporins in human tissues. They're present in all life forms, microorganisms, plants. Aquaporins are the basis of water movement in all of these species. So teaming up with Søren Nielsen at the University of Aarhus in Denmark, we solved how kidneys can concentrate water. And I won't go through the details, but this is what allows us to filter 200 liters of primary urine every day. Our plasma is filtered again and again to release the toxins, but reabsorb 99% of the water. Defects in this cause problems such as bedwetting or severe problems with people with defective renal function. Another protein of the family, aquaporin 0, is present in the eye, in the lens, where Masato Yasui from Tokyo established it as the cause of congenital cataracts in small children. The presence of the AQP4 protein in brain provides an explanation for the maintenance of homeostasis in brain. And a defect in this causes problems such as brain edema. And our colleagues in Norway, led by Odla Peter Ottersen, now the rector at the University of Oslo, and his student, now a faculty member, Mahmoud Amiri, showed that the normal presence of this protein in mouse brain, and if you look at the middle sections here, you see what happens after a defining brain injury. Defects lead to edema and infarct of brain, but mutant mice, engineered so that their water channel proteins are mislocalized, are partially resistant to this. This gives us hope that the problems of brain edema, which are very common following closed head injury and stroke, can be managed by medicines that inhibit or mislocalize AQP4. And AQP5 is present in secretory glands. They cause the release of water in tears, sweat, and saliva, something we all encounter. Now, the glycerol transporting homologs are present in a number of tissues, particularly in the basal levels of skin. What you should see here is an intense staining in prenatal and postnatal mouse skin, the presence of the aquaglyceroporin 3. And we didn't recognize the importance, but the beauty industry quickly got onto this. Now, I should point out, I have no financial ties to Christian Dior Company. I would love to have financial ties to Christian Dior. But when two of their executives came over to visit me and invited me to give a lecture in Paris, it was clear that the strings attached were something I couldn't accept. But just for your information, their chemists have identified some natural molecules which you can purchase in this expensive skin cream. It's quite expensive, about 50 euros for a 50-gram jar. And apparently, if you use enough of this, you start looking like this. I'm not sure that constitutes scientific proof. But I saw this in the back cover of the Marie Claire Beauty magazine at the Charles de Gaulle Business Lounge. And I did the honorable thing. I ripped it off and put it in my briefcase. And those of you who speak French will see that they had some pretty bold statements. That this skin lotion causes an increase of the aquaglycerin 3 with spectacular results. And what's this? The Nobel Prize in Chemistry. I showed this to my mother. Mother is 80 years old. She's a farm girl. She never went to university. My dad was a college professor, but he died some years before this. And I showed this to Mother. She smiled. She said, "Peter, I think you're finally doing something useful." Our mothers know us better than we know ourselves. But I think some of the work might be useful. Finishing the story here, aquaglycerin 7 is present in fat. And if you think for a moment, our fat is designed not to make obesity, but to provide an energy source when we're fasted or starved. And the release of glycerol from fat occurs through aquaglycerin 7, where it's taken up through the liver and through aquaglycerin 9 and is converted to glucose. So we can efficiently maintain body glucose levels by burning fat during fasting. And this is interesting because it's also the portal for the entry of arsenic into and out of cells. A surprise observation made by Barry Rosen and his team in Michigan established this, and explains the the toxicity of arsenic and the clearance of arsenic from mammalian species. Of course, the prevention of arsenic toxicity from contaminated groundwater, the prevention is the provision of pure drinking water, which is a basic human right. Plants have aquaporins, and shown briefly here, this is work of a German scientist, Rolf Kaldenhoff, who was at the time at Würzburg. A normal Arabidopsis can maintain foliage with a thin arborization of rootlets, but a mutant in which the rootlets have a reduction in aquaporin family members will maintain water uptake by sending out an arborization of rootlets, each rootlet having less water uptake, but the plant compensates. So that's a little background on the aquaporins. Now, 9 years ago, this month, I got a call early in the morning from a friendly voice with a Swedish accent informing me that I would share the Nobel Prize in Chemistry. Now, chemistry, I was actually not a very good chemistry student. I decided to keep that information to myself and listen politely as he explained they would have a press conference in Stockholm, and in 10 minutes, the entire world would have my telephone number. I ran to the shower to get ready for my day, and my wife, Mary, always organized, always calm, called Mother back in Minnesota to share the news. Mother thought for a moment when she was told I would share the Nobel Prize in Chemistry. She said, "Mary, tell Peter that's very nice, but don't let this go to his head," reminding me that it's still important to do something useful. And I think that's the value of science. We don't always know where the science will take us, but there are useful applications of the work that oftentimes go far beyond our initial objectives. And here we were on the stage in Stockholm, which was a wonderful event, and any resemblance I had to Alfred Nobel ended when I took off this mustache. I resembled an individual I won't mention to you who was an unpopular ambassador to the United Nations. Ambassador Murphy knows exactly what I'm talking about. And at age 55, I shaved off the mustache, and my wife said something wonderful. She said, "Peter, you know, you look younger without that mustache," which I considered good news. So this is the prize, which is a nice event, but the real value is not who gets it for what, but that the entire world gets news on the first week of October that science is doing something special. And in December, when they award the prizes, the world is again reminded. And it's not just physics, medicine, and chemistry, it's literature and peace, perhaps the most important prize of all. So this was an event which I was very pleased about, but it was clear to me that I had other adventures that I'd long foresought and made some changes in the direction of my career. And the notion of doing world health, which was my initial objective as a medical student, became an opportunity when Dean Clagg offered me the directorship of the Johns Hopkins Malaria Research Institute at the Bloomberg School of Public Health. And this institute was made possible by a magnificent gift of a private individual, initially anonymous. Now it's public. It's, in fact, Michael Bloomberg. And the orientation of this institute is to provide a basic scientific approach to the study of malaria in the laboratory with the application of the discoveries in the field. And of course, as all people in World Health recognize, while malaria is improving in some parts of the world, it's still a dire epidemic in many places. So the current world malaria map shows that the great bulk of malaria, of course, is present in sub-Saharan Africa, including Zambia and Zimbabwe, where we're currently working. Now, there is an aquaporin twist. Red cells have an aquaglyceroporin. That's how glycerol enters red cells. And of the challenge of malaria, where the parasite resides within the cells, the parasite can grow and divide, synthesizing glycerolipids by taking glycerol in through aquaglyceroporins of its own design and the host's own aquaglyceroporins. So our hope was that this could provide evidence that would provide for new therapies for the treatment of malaria. And in truth, the aquaglyceroporins are important. They're probably not sufficiently robust to make new medicines. But that's science. You check and you move along. But being committed to the malaria field, we pursued this series of studies. And just for your background, those of you who are not hematologists, here are red blood cells. Here's one invaded by a single parasite. And here's the trophozoite. This is when this cell has divided to 32 daughter cells. And if you imagine this cell breaking, the release of 32 cells, 2 days later, 32 cells are now infected, and there'll be 32 times 32, 1,000, 2 more cycles, 1,000,000, 2 more cycles, 1,000,000,000. And the individuals infected with malaria have grams of malarial sludge passing through their circular systems, causing horrific fevers and end organ damage. The mosquitoes that carry the malaria parasite are also dependent upon aquaporins for water balance. You may not think about it when you're bit by a mosquito, but the mosquito needs to fly away. And in order to jettison excess weight, it uses fluid through the Malpighian tubules of aquaporins. The net result is the mosquitoes bite and infect young people. The great bulk of malaria, of course, is in children, and particularly the children of sub-Saharan Africa. Now, these cute little boys who are working outside the village of our research institute in southern Zambia is probably, if each had malaria at least once, and in previous decades, they would definitely have had malaria, and maybe some would not survive. But you can see with good medical care and of particular importance, the provision of prevention of malaria, these little children, while not rich by Western standards, are happy. They're having normal lives. Now, this slide was provided by Dean Clegg, and it's an important issue because while we know that up to 1 million children die of malaria every year, it's quite clear that of the survivors, many do not escape entirely healed. This little boy was brought near death to the mission hospital in southern Zambia in a coma with cerebral malaria, and his life was saved by Philip Tuma, the doctor on the far right, a Johns Hopkins pediatrician who's devoted his entire professional career to the treatment of Zambian children. And the child's life was saved, but you notice he's left with a disconjugate upward gaze. It's because he's left with cortical blindness. He will never see again. So in addition to the loss of life from malaria, there's a tremendous morbidity. And in the third world, life is difficult. But when you're left with a handicap, it's very much more difficult. So here is the logo for our malaria team. And here are some of us at our field station in southern Zambia. Shown on my right is Dr. Tuma in the sunglasses. On my left in the blue shirt, Sungano Marakura. He is the scientific director of our Malaria Research Institute. And he's a very interesting and wonderful person. He's a Zimbabwean national, educated at Oxford and the London School of Hygiene and Tropical Medicine. And his ambition is to do something for his people and return to his country. So we really have an outstanding team with our other Zambian workers at work in Macha in southern Zambia. Now, we are a research team. We are not there to provide health care for the entire southern Zambia. But in pursuing the problem of malaria, doing field surveillance, a very wonderful and positive event has been forthcoming. this represents, and it's a little complicated, but this represents the 11-year clinical experience in the Mancha Hospital, where for years and years, every year, about 1,500 to 2,000 children age 5 and younger would be brought to the hospital with between 30 and 70 deaths. In 2003, the introduction of artemisinin in combination led to a major reduction in the deaths and the prevalence of the disease. Notice, down from 1,700 to 400, deaths down to 18. And it declined still further. 2006, the year there was a stock outage, the medicine became unavailable, the disease returned, but then it was reduced again when the medicines became available, and insecticide-treated bed nets were provided. And there are many, many organizations, including faith-based organizations, as well as the President's Malaria Initiative, that provide these bed nets, and reduce the prevalence of the disease still further. And in fact, last year we were down to just 2 deaths. 2 is still unacceptable. We're not at 0, but good things are happening. So it's a bad problem, but we think we know the answers of how to provide it. But there are difficulties. Some of the difficulties are logistic. These subsistence farmers do not live in cities with bus lines and taxis. They live in the rural outback. And this was a photograph just taken of the path to one village just at the beginning of rainy season. By the end of rainy season, the grass will be 2 meters tall. And in these rural domiciles are perhaps the most innocent people on earth, working as subsistence farmers to provide medicine or to provide the food necessary for the families. And so our surveillance team would follow up on cases of malaria and check for the ever-present subclinically affected individuals. A large number of people who've had malaria will carry very small burdens of the disease and are unaware that they're infected at all. And of course, they are the source of the return of malaria from areas where everyone has been treated. And they have enormous trust because of the long-term commitment of Dr. Tuma and his colleagues in southern Zambia. But as you can see, they work hard and their lives are not easy. The children work alongside the adults. They also know how to play. These kids just love to have a good time. And there's great appreciation for children in sub-Saharan Africa. Although these are amongst the poorest of the poor on the planet, there's great reverence for children. Children are treated as well as it can be. And it's really, I think, the focus of families. And they respect education. You can't see it, but the little girl on the left is holding her school book. And upside down it says, "School is cool." Now, one of the problems, of course, with malaria is that there are regions where malaria can be brought under control and regions nearby where it is not under control, often because of geopolitical difficulties. Some of you may recognize this bridge at Victoria Falls separating Zimbabwe on the far bank and Zambia on the near bank. And Zambia has had, because of a number of good things, including good governance, a significant decline in the amount of malaria. Whereas in Zimbabwe, things have not gone so well. And just for your interest on the map, we're studying malaria in southern Zambia. This is the Mancha site I talked about. In northern Zambia, on the border of Congo, where malaria has never been under control. And in eastern Zimbabwe, where malaria was under control, but it has now escaped control. And this is Robert Mugabe, the president of Zimbabwe, who we must recognize and respect as the liberator of Zimbabwe. But the great problems with inflation have led to a great decrease in the prevalence of medical supplies and public health administration. And this once rich country is now in many regions riddled with malaria. They have the resources, but they don't have the coordination. And children like these develop malaria. So with the grant from the National Institutes of Health, we're currently working in these 3 sites, including eastern Zimbabwe, where a medical team such as this group here, shown along with Sungano, is on duty, and we hope to provide better access to the medicines and the diagnostics to bring the malaria epidemic under control again, and we hope eliminate it. And of course, the clinics, while they're there, they're overwhelmed with a burden of disease. The Hunde Valley in eastern Zimbabwe, which had no detectable malaria 20 years ago, has more than 100% of its citizens affected every year. People like this little child, and also pregnant women are badly affected by malaria. This grandfather would like to see his grandchildren grow up without malaria. And they're very religious people. They're very committed to their lives. They have names that are religious: Isaac, Miriam. They also have names that are really interesting in eastern Zimbabwe. Give more, love more. I met a man named Nevermore. I asked him, how could you be named Nevermore? He said, well, my mother had 9 children and I was the 9th. I was born, she said, Nevermore. I think we owe these people everything we can provide. And here's Sangano in front of the National Institute of Research in Harare, looking forward to returning to his country of origin to take up the fight against malaria. Now, Mike mentioned that I'm involved in some science diplomacy efforts, and I'm going to just go very quickly over these, just because this is another, I think, benefit of the work we do as scientists. There are global doors that open to us that are not open to, oftentimes, the politically elected individuals. And I'm just going to talk about our work in Cuba and the Democratic People's Republic of North Korea. We've also been to Myanmar, Burma, Tunisia, and in June I was in Iran. And in all of these locations, I think there's reason for optimism, but I'll just talk about Cuba and North Korea briefly. So Western Europe, of course, has recognized Cuba as an independent, sovereign nation. In the United States, we still have a big political problem. And it's been 50 years in the making, and we have an adversarial position towards the Cubans. In many ways, this allows, because of the trade sanctions, the preservation of old vehicles, which we all think of in nostalgic terms. And the people in Cuba are very welcoming. Here's a fellow who was hoping I would smoke that cigar. I'm sure it would have killed me. I gave it back to him after the picture was taken. Now, well, where investment has been made in Cuba, the architecture is well preserved, and there are many good things to say. But where there has not been investment, it's crumbling, and it's quite clear that our sanctions, while well-intentioned, have had some negative consequences. But there has been a constant investment in science in Cuba, in post-revolutionary Cuba. Shown beside me is the director of the Cuban National Biotechnology Lab on my right, and the director of the Finley Vaccine Institute on my left. These gentlemen are hard at work because it's been a priority in post-revolutionary Cuba to prevent and treat disease of children. And this was, in fact, one of the reasons for the revolution. And this is, of course, El Comandante Fidel Castro, who we spent an evening with. And it was quite interesting. There are a lot of critical things we could say about human rights in Cuba, but healthcare has been made a priority in Cuba. And I think there are things that we can agree on and things we can learn about vaccine development in Cuba. The University of Havana has been open ever since the revolution. But it's not peopled by old scientists like myself. It's peopled by young people like this. I lectured at the University of Havana and was swarmed on the podium by these young Cubans. They're in their late teens and early 20s. They want a chance to do science and get involved in global health like we're involved in. They will join us if we make it possible. In closing, I'll talk about briefly about a series of trips we've made to North Korea. North Korea is often referred to as the Hermit Kingdom. Probably the farthest place we can go from normal relations. And we've had a series of trips to North Korea, shown here with one of the directors of their microbiology laboratories in Pyongyang is our team from the United States. But visiting North Korea is a little bit like visiting the Twilight Zone. This was a television program back in the 60s in the United States. It looks real, but there's something not real about it. Where are the people? This is downtown Pyongyang, Monday morning at 10 o'clock. Something is not real here. And of course, the authoritarian leadership provided by Kim Il-sung and then his son Kim Jong-il, and now Kim Jong-un, the grandson of Kim Il-sung, is seen as the absolute word in all matters related to North Korean policy. And they have a notion that they will be self-reliant, juchi, they will go it alone. But in truth, they cannot go it alone. North Korea cannot feed itself. It's a desert climate, and due to inadequacies in the agricultural systems, they are given food by the West, which we hope will feed the peasants, but unfortunately oftentimes gets siphoned off to feed the army. They're constantly reminded of the need to work hard for the regime. But there's an element of optimism, and the optimism has to do with science. I'm shown here with one of our members of the National Academy of Sciences of North Korea in the grand reading room. And what should be on display there but the American publication Science magazine. And scientists in Korea are oftentimes younger people. They are often members of the party because they're forced to be, but their real motivation is to make the world a better place for their children and their grandchildren through science, through plant biotechnology. And this is the director of the State Academy of Science. John, you might recognize that tie, Mike. I broke the rules. That's our Johns Hopkins tie. It's the one I wore when I gave my lecture in Stockholm, and I gave it to him to give to a young North Korean who gets the first Nobel Prize from North Korea. So the government will change, and we can't do much about it, but there are ways we can engage with the North Koreans. And one opportunity is the Pyongyang University of Science and Technology, a new campus built on land outside of Pyongyang due to the magnificent gift of this man. This is James Kim, a businessman, Korean-born, who has made quite a substantial amount of money in Florida, and has built this campus to teach young Korean scientists English, and he hopes bring them closer to the rest of the world. But it's a difficult challenge. Here are the students at the Pyongyang University of Science and Technology on their way to class. I guess we would say this is not a usual kind of party school in the United States, unless you talk about the Communist Party. They maintain strict discipline. When I lectured to them, I started out just to see how good their English was. So I told them a joke. It's a pretty good one. I'll save it for another event. Nobody laughed. So I said, that was a joke. That's very funny. And they all burst into laughter. They need permission to do what we would all spontaneously do because of fear. And even when speaking to us, if you look over the shoulder of the gentleman on the left, he's being observed at the same time. So there's fear, but there's, I think, reason for optimism, even in North Korea. And in closing, I'd like to tell you about this gentleman. This is Rong-gi Hong, the director of the International Agency of the North Korean Academy of Science. Excuse me. Dr. Hong has been to Europe, to South America, to Canada, and now to the United States. And we became pretty good friends. And I asked him if he'd ever had the opportunity to visit South Korea, the Republic of Korea. He's a little older than I am. He grew up during the Korean War. And when I asked him this question, he became very quiet. With sadness in his voice, he said, "No, so near, but still so far, it is not possible." But we became good friends, and I found him to be a really wonderful colleague. He's a member of the party. There's some clear differences. In the last morning of our week in Pyongyang, of our first trip, we shared a breakfast. And at the end of the breakfast, he told me, the children see Americans as adversaries. When he told his grandson, who was 6 years old, that he would be staying in a hotel in Pyongyang with Americans, the little boy became very alarmed. He said, "Grandfather, bring the rifle." Afraid of what we would do to him. Now, I carried a box of granola bars just in case I should need a snack. I had not opened it during the trip, and I thought, well, Dr. Hong, why don't you give these granola bars to your grandson? I never thought more about it. A year and a half later, when we met with the North Korean Academy of Sciences at the Carter Center in Atlanta, Georgia, we started our discussions with a very formal kind of negotiated phase. A table like this, we were on one side, I was at the center, North Koreans on the other side, across from Dr. Hong. And he delivered a very stern message. We have not received any of the rewards promised. We have not been able to initiate any of the collaborations. Our party officials feel that this is all a waste of time. Pretty grim way to start a meeting. And now it's my turn to respond. So I said the first thing that came to mind, I said, "Dr. Hong, how's your grandson?" He smiled, and I said, "Did he tell you to bring the rifle?" He said, No, but bring back some candy." Maybe that's what science diplomacy is. One person at a time, a grandfather and his grandson. Maybe we can make connections in a special way. I know we can. We're doing it all the time, every one of us here. So as the young scientists, and I see quite a few young scientists here, I'm glad to see you. As you get older and you look back in your careers, you'll wonder, what did I give up? And I dare say you probably feel, well, we gave up material rewards, but we gave up nothing that was important to us because we had an opportunity to do something very permanent for the world. So I'd like to close with a little wisdom here from the Mandarin. In Chinese, the word for crisis is 2 characters. In Korean, it's very similar. In Chinese, Wei-ji, in Korean, it's Wei-ji. And the first character, Wei, time of danger, Ji, time of opportunity. Think about it for a moment. Danger in the world, epidemics, malnutrition, climate change, wars, bombings, many dangers out there. But the opportunity through medical science is a way to open the doors to make the world a better place, and I know we will continue to do so. And with that, let me thank you for the opportunity to be with you here today in Berlin.